Chapter 7, Section 13, Ribose Switches. Now, you've been taught from early on that a lot of the work being done, most of the work, all of the work being done in the cells is due to enzymes, enzymes being polypeptides, protein-based. Well, it turns out that no, not all the work. Majority of it, yes. But some of the work's going to be done by RNA molecules. I mean, think about it, ribosomes, you know, the thing that actually makes the polypeptides that will become the enzymes. Ribosomes are primarily RNA, a little bit of polypeptide in there just to hold the RNA parts together. But it turns out that there's more than just these ribozymes, these RNAs and ribosomal RNAs, things like that. There's this new thing that we're getting into, that we're studying in genetics and cell biology, that is referred to as ribose switches. Okay. When you were taught intro cell and molecular, maybe cell biology, RNA, you were thinking RNA, just this one strand of RNA. Okay, you got more into cell biology, then you get into genetics, and then we start to teach you that, okay, that strand, that just string of RNA, that two-dimensional string, is very unstable. So what happens is that unstable two-dimensional strand will curve back on itself, complementary base pair with itself, forming these three-dimensional structures. These three-dimensional structures that are referred to as stem loops. These stem loops can have even more levels of structure to them. Having this complementary base pairing on itself, forming these stem loops and bulges and things, gives these RNA strands some stability. It's also, over billions of years of evolution, has also become a mechanism to recruit in ribosomes and transcription translation factors. That's where these ribose switches come into play. Positioning of these stem loops is very important. In one position, the stem loop could aid in the recruitment of the large and the small subunits of the ribosomes. But in a different position, that stem loop would interfere, stop translation. So we're now looking at these stem loops and the terms of what are now referred to as I said, these ribose switches. They're centered around regions that are referred to as aptamer regions. This is a region that allows for a stem loop to occur. There is multiple regions for complementary base pairing. One is a dominant, or a recognition that allows for the ribosome to bind to, may even, as I said, aid in recruiting. But when it goes to an alternate position, an alternate region that can also complementary base pair, so that stem loops moves over. Well, now it interferes. Now translation is not allowed to occur. You look here. Here's examples A and B. You look just here at A. There are three regions, 1, 2, 3. 3 RBS, ribosomal binding sequence. This is where the ribosome's the small subunit's gonna come and then translocate to the first codon where the initiation tRNA is. This is the aptamer region. Here's the stem, here's the loop, hence its name, stem loop. 1, 2, those are regions that complementary base pair with each other. There's long strands. Here they're just showing it like what, 5 nucleotides in length? No, it's going to be much, much longer. But something about this region means it has a high affinity for a product. What product? Well, chances are it's the product that the protein here that's going to be translated here is responsible or part of the metabolic pathway to make. Remember, feedback inhibition? Well, here's the final product that comes back and will interact with this aptamer region. By interacting here, it pulls apart this stem loop, allowing for region 2 now to interact with region 3. Hey, look, that's supposed to be the place where the small subunit comes and starts to interact. But right now, it's part of a stem loop, which means no small subunit, thus no larger subunit, thus no translocation, no translation. All because feedback inhibition. Well, think about it. This is a good thing because this mRNA is not being degraded. It's not going away. It's still there. But you have enough of the final product. So why keep making enzymes for a metabolic pathway you don't really need right now? So there you go. You have a high enough product, high enough concentrations in the cell, means it's now going to go and interfere with making more enzyme that would be responsible for making more of the product. Okay. Protein enzyme turnover, enzyme levels drop, which means given enough time, this metabolite level will also drop, which means this goes away, and turns out 1 to 2 is more stable than 2 to 3. So then it shifts back to this, and ribosomes can go back to doing their job of translation of this coding sequence, this gene or this operon. You see it's quite similar here, but a little more dynamic. This expression platform, as they're showing here, this is where the ribosomes and any translation initiation factors and whatnot will collect. Here's where the ribosome binding sequence is. But again, you have the difference between A and B here is region 3 is not part of the ribosomal binding sequence. Here it is, here it's neighboring. But still, you get this shift from 1 stem loop to 2 stem loops because of a metabolite, a product of the pathway, which interferes with the ribosomal binding site. Either the RBS becomes part of the stem loop, or it's now so closely associated with the second stem loop that the small subunit, large subunits just can't get access. So as I said, that expression platform, that's where everything's going to collect. Small subunit initiation, any initiation factors that may be there, any proteins that are going to help regulate, yes, no, can this translation event occur? So not only do you have outside proteins, activators, initiators, regulators that can bind to the messenger RNA, now you're going to have the messenger RNA itself is going to be regulating expression off of it. It's actually going to be regulating at the translation level as opposed to, a outside protein coming in and binding. We see this with a few metabolic pathways, the pathways for thiamine, tetrafolate, hydrofolate, which we know as folic acid, vitamin B12 synthesis. So many of these you see here can go back and turn on, turn off a ribose switch. If their levels get high enough in the cell, they go bind, change the stem loop, secondary or three-dimensional structure, interfere with. Their levels drop. They no longer associate with the messenger RNA, and then the Three-dimensional stem loop structure goes back to the original. Ribosomes can now interact. Ribosomes can now make. We also see this control at the transcription level. Past few slides, I've been talking translation. Access by the ribosomes, allowed or interfered with. We also see these ribose switches coming into play at the transcription event. Because remember, as the RNA polymerase is marching down, or I should say whizzing down, the double helix of the prokaryote or the fungi and some of the plants, the messenger RNA is not coming off as a strict secondary structure. It's coming off, and as more and more of its length is coming out the RNA polymerase, it automatically starts forming those stem loops. Well, if the metabolite is at high enough concentration, regardless of what the transcription factors are saying to do, there's high enough concentration in the cell, the metabolite is going to bind to that growing messenger RNA chain, start forming, alternate three-dimensional structures that will interfere with RNA polymerase activity, will actually cause premature termination. The entire gene will not be transcribed, or the entire operon will not be made into a polycistronic messenger RNA. How it actually forces this, don't know, don't care. It just does. All right. If you really, really want to know, ask Dr. Albie in genetics class how these ribose switches can actually force this to occur. As I just mentioned in the last slide, we see these ribose switches and their control of translation and transcription, mostly in bacteria, archaea, few plants, and fungi. Not all bacteria can do it, not all archaea, just like, not all plants or fungi, but a select few. They think it's in a remnant for some of these older species that have been carried over, conserved evolutionarily from the RNA world, back before there was DNA, back before there were proteins and, life, quote unquote, life as we know it at the cellular level. RNA was king. Before there were protein polypeptide-based enzymes, there were ribozymes. So they think that these ribose switches are a holdover from those original RNA/ribozyme world.